collaboration across borders.
Hello, everyone. Welcome to this session of the Assemble Plus conference. So we're going to have a series of talks this afternoon, and we are going to start with Pedro Martinez from the University of Barcelona, and he's going to talk about deconstructing the enigmatic Oisilomorphs, which must be a very interesting group. So Pedro, go ahead. Thank you very much for accepting to be part of this, and it's your word now.
Okay, very good. So thank you, Assemble. Thank you, Adelino, for the introduction and for inviting me. Let's see, how this thing works. I'm not so good at, okay, let's see.
So you have the PowerPoint open and then you share screen, click on share screen.
Do you see it?
No, you click on share screen first.
Okay, good.
And then you choose the file. You choose the one that is open.
Share screen, okay. Let's see if it's sharing. Same. Is it working now?
Working fine.
Very good. Okay, so first part of the talk done already. So anyway, what we're going to be talking about today is, well, a fraction or a few things of what we do in the lab, or what we've been doing during the last few years. As you know, I'm working at the University of Barcelona in the Department of Genetics, and the main interest of the group is the evolution of animals. Or let's say we are working within the discipline of Evo-Devo, and our main focus is on trying to understand the origin and diversification of bilaterian animals, okay? Our models are a group of animals which have this strange name of Oisilomorphs, okay? So let me see, tell you exactly why are interested in the origin of bilaterian animals, and this is for a rather simple reason. You will see it here. Let me see. It doesn't move to the next slide. Okay, something missing. Okay, so here is this graphic encapsulates a little bit the interest of the question we are posing, which is how the bilateral animals became so successful. You have in this very simple graphic a distribution of a species, or if you wanted, a distribution of morphologies of both living and extinct species. And the first thing you realize, if you look at this clear color, is that it's about 98% of the species living or extinct are bilateral. They have bilateral symmetry, and only a small fraction have other kinds of symmetries, radial or no symmetries at all. No? So the question we posed at the very beginning, maybe like 15 years ago, when I arrived to this my new lab, is how come or why do bilaterian animals have been so successful across all kinds of ecological niches, okay? And so the reason is essentially derived from their morphological innovations. They have innovated, or they have incorporated some innovations who have been key to their success, to future success. And these are essentially three kinds of innovations that came with the origin of bilaterian animals. One is obviously that the invention of an orthogonal axis. So you have a left and right sides of your body, which are mirror images of each. Then it was the mesoderm was an invention which is for instance, muscles, but also circulatory system, etc. And a third invention that came with the origin of bilaterian animals was the presence of a centralized, an aggregated system of neurons at one side of the body, which we will call a brain, okay? And then you will say, okay, well, why is that relevant? I mean, what has all of this to do with chain oisilomorphs? Huh? But then you will understand this through the seminar. The most important thing is upfront telling you what is a chain oisilomorph, right? And what do we need to know if we really want to understand these creatures and how do they inform us about the origin of bilaterality. So this strange or enigmatic group of animals, so it's, sorry, I think I'm missing here some, okay, but anyway, I think I missed one slide. Anyway, so in order to understand what are chain oisilomorphs and what do they provide us in terms of informing the origin of bilaterian animals, I will subdivide this short talk in three different topics. The first, I'll tell you about chain oisilomorphs themselves, the phylogenetic affinities, where do they sit in the tree of life? And then I will tell you a little bit about how they are working on in terms of like characterizing their genomes, the gene families, and how do these genes influence the growth and differentiation of different tissues. And then at the end, I will talk about some specific organ systems that are relevant or very important to what we do. Let's start by the first one. So what are chain oisilomorphs, okay? And what are their phylogenetic affinities? Okay, chain oisilomorphs is a monophyletic group of animals. Okay, it's which is composed of three major clades. One's called cenoturbelids, another one's called nemertodermatids, and a third one called acils, oisil flatworms. These two last ones form what's called the Oisilomorphs. Okay, and with cenoturbelids, obviously, chain oisilomorphs. These are normally rather rare animals. So except some acils, for all these other two groups, we have identified so far very few species, okay? New expeditions are identifying some cryptic ones, okay? But these still are not fully described species. Anyway, so the only species-rich group is the acils, for which we know approximately 400 species. Maybe a dozen nemertodermatids and a few 6 to 7 cenoturbelids. Besides that, they are very rare in terms of numbers and hard to find. In any case, the important thing is all of those belong to a monophyletic group that some people call it phylum. Where do they sit in the tree of life? Okay, there are two competing hypotheses. Okay, and that's why I said I put at the top the position of chain oisilomorphs as today. Okay, so tomorrow may be a little bit different, but in any case, most of those phylogenomic studies have can be divided. I mean, have given rise to two competing hypotheses. Let's say that the one that is mostly accepted would be the one to the left. Okay, in which chain oisilomorphs are the sister group, okay, of the remaining bilaterians. So in a sense, it would be the first group of bilaterian animals, okay, and the only one, okay, of early bilaterian groups which is still extant, alive, no? There is a competing hypothesis, and this depends a lot on discussions of models of evolution, etc., which puts the chain oisilomorphs as sisters to the deuterostomes, okay? So it will be like a deuterostomian group, okay? But for now, let's imagine that as many people think that the left topology is the one that represents best the position, say, Oisilomorphs. As you can see, being basal or being the group sister to the remaining ones should be good proxies of how an ancestral Bilaterian group may have looked like, okay? And in that sense, understanding how Oisilomorphs are built at both genomic and morphological levels should be relevant in terms of understanding how the primitive or the older groups of Bilaterian animals may have looked like. Okay, so this is with respect to their position and why they are relevant. In our laboratory, we work with different models within this Oisilomorph group, okay? And the models we are using essentially are very few, okay? Because of difficulties of getting enough animals, etc. So we mainly try to use one member of the Xenoturbelids, okay? Let me go back to this. So we are working with one member of Xenoturbelids. There's one species called Xenoturbella bocchi. You will hear about it a little bit later on. And then we are using mainly one species of acils, which is this one with the green color, which is called Simsagitifera roscofensis. It doesn't really matter the name of the species because they are complex, but just think that we have one Xenoturbelid and one acil in the lab, okay? So the model species that we are using in the laboratory, obviously there is not a mouse or Drosophila, we don't have those tools, but essentially are those ones. Within the Xenoturbelids, we are using this strange animal, no? It's more like a sock, no, a little bit sort of like a boring animal, okay, but interesting from the evolutionary point of view. And this is this animal, Xenoturbella bocchi, which is normally collected in Sweden in a fjord, which is called Gullmars Fjord, is the fjord which is in front of Kristineberg Marine Stations. For those of you who have been traveling there, they normally range in the order of 1 centimeter long, okay? And it's really hard to get, okay? Normally you get them through dredging, okay? Dredging in the fjord, sand in the bottom 50 to 100 meters, okay? So it's a lot of dredging and a lot of mud sieving. And then if you are lucky, you get out of like many kilograms of sand. Perhaps if you are lucky, you get a few Xenoturbelids or Xenoturbella. At least in our hands, I mean, it's very hard to get them. So if we get like 10 animals, okay, then we consider ourselves very lucky, okay? I see that around here is also Ulf John Dilius. Maybe he's luckier than us when he goes and collects. But in our case, I mean, we've never been particularly lucky in terms of numbers. Xenoturbella will look like this. This will be a cross section. So you can see that essentially it has like a gut in the middle and then a gut cavity. And then outside you have epithelia and a lot of like a neural neurons, which are essentially lying in this yellow part, okay? This is, it's relevant in the sense that we use this as a guide to study the morphology of those animals. What lab models, what acils do we use? I told you we are using 2 acil flatworms. One is this one, which is collected here in Brittany, France. It's called Simsagitifera roscofensis. And this is easier to collect because normally all of these patches in green are full, are thousands of millions or millions of animals like this, okay? So at low tide, you can get in the areas of, in Brittany, you can get tons of those animals. So in that sense, it makes a more suitable system than Xenoturbelids, for instance. You can get millions of animals. So if you want to do large scale studies, that's possible. These animals, we find it in Roscoff or in the area of Brittany, but you can also find them in Normandy, in Portugal, in northern part of Spain. So most of the eastern part of the Atlantic, they are there. The advantage of these animals is, as I told you, you can get, well, we'll skip that one because I think it's not that relevant, the general morphology of acils, but we get these, normally they have a few millimeters long, as I told you. These animals can be found at low tide in many areas in Brittany, okay? And then here you see collecting animals in little pools, okay? And these little pools, actually, if you blow up the picture, then you'll see there is thousands and thousands. And of course, since we have to always wait for low tide, meanwhile, well, we have like nice restaurants in the area and good drinks while we wait, okay? Here is a simplified look at the development and morphology of those animals. Here you see an adult animal in A1, okay, few millimeters long. In the pools, a close-up of the pools, you will see all of these little worms. The lighter patches in the middle of the animals, these are eggs, so these are gravid animals. They are all hermaphroditic, okay? Normally, during the winter, they will be spawning eggs in form of cocoons, 15 to 20 embryos that are synchronized inside each cocoon. And eventually when they hatch, they will get the algae that makes them green from the environment, okay? So all of these little dots in here, in the figure are algal symbionts, okay? Very good. So this with respect one animal. This animal is normally collected, but never, or we don't keep it in the lab for long periods of time, are difficult to collect. There is another species which originally comes from the northern part of the US, here in the coasts of Maine and Massachusetts. And this one, we and others been able to culture them in the lab. Here is our department in Barcelona, here's our students. This can be cultured on a regular basis. What this means is that you can close the cycle and you can have easy access to all the developmental stages at any time, okay? There is further advantages that we've been using, for instance, the fact that you can do knockdowns using RNAi technologies, et cetera, okay? So which has allowed us to study gene expression and gene activities by testing their functions. Okay, this is a parenthesis. I won't explain you many details of the data because I think for you, since we have like an audience with different backgrounds, probably you want to know exactly what do we do and what kinds of animals we are exploring. So these are the models, as I told you in the first point, that we are using in the lab, okay? And these correspond to two major groups of Oisilomorphs. Remember, that are probably old bilaterian animals, okay? So of course, if you want to know how do these animals function or are constructed, you need to have access to the genes that make the animals, no? This means understanding the genomes, okay? And to understand the genomes, understand the genomes of as many animals as you can within this group. This have been done. At present, we have partial genome data from 6 different animals within this group, okay? There are partial genomes from Cenoturbella, a couple of nemertodermatids, and then a few acils. But for 2 of those, for Cenoturbella and for Sagitifera, okay, we have pretty complete genomes, okay? Partial genomes have been already published, okay? There is a paper in Current Biology 2019. You can check if you would like to know the first draft of those genomes. Okay, so here are some characteristics of these 2 genomes. Cenoturbella is a rather compact genome, while Sagitifera, well, that's pretty big. It's about half of the size of a human, okay, genome. Cenoturbella is still relatively poor in terms of like sizes of scaffolds. Okay, this is built with the scaffolds which are relatively small, 5 to 10 kbs average, minimal. But Sagitifera, we are about to publish a new genome in which we have an average scaffold, what it's called N50s, above 200 kbs. Okay, and we're starting to work with high-phi sequencing, so eventually we'll get like the 2 chromosomal size. So for these 2 animals, we have pretty complete genomes, which means that this has allowed us to study different gene families, composition of gene families, and the expression of some of those gene families, okay? Which have informed us about like the construction or the development of particular tissues, like the muscles and neural systems. The papers are out there, and then I won't go into the details, but just to tell you that, I mean, this has provided very important information per se, so you can understand how the architecture or the composition of those genomes change over time, okay, by comparison among them and with respect to other bilateral or radial animals. But on top of that, this has allowed us to generate probes for like understanding gene expression of many different genes. Then of course, I mean, if you really want to understand how these, the morphologies of those animals have an impact on or inform you on the structure or architecture of like an old bilaterian, what you have to do is to describe the organ systems in good detail. This has been done over the last few years using conventional and more sophisticated methods, okay? First, and the most important organ system for us is the nervous system, because we want to understand how this cephalization process, this construction of compact brains that accompany, that were happening in concert with the origin of bilaterians, how these things have happened. And the chain Oisilomorphs are very interesting because the following reason. And this is, if you analyze, these are different Oisilomorphs. Here you will have Xenoturbella, the Xenoturbellid. Here you have some nemertodermatid, and these are different acils. The diagrams in blue are different architectures of their nervous system. So you can see in Xenoturbellid, essentially you have nets of neurons under the epithelia, nothing else. And progressively, if you wanted to call it like this, you have in some members of the acils really compact aggregated sets of neurons and everything in between. So Xenoisilomorphs are very interesting for studying the evolution of nervous system because you have all kinds of architectures from simple nets to compact brains represented within this phylum. And because this reason, we wanted to further study how these nervous systems are constructed, okay? Because again, you see here the first examples probably of compact brains in for any bilaterian animal. So we use it conventional, okay? So this is conventional, here you can see the nervous system of one of those species, okay? Here with the aggregation of brains, and these are nerve cords, and then you have like a net of interconnected neurites, okay? This is from the dorsal and the ventral side of Isodiametra, but also you have combinations of antibodies that allow you to identify specific kinds of neurons here in green and in magenta, and then you have also muscles in blue. We have here been able to detect detailed structures of the brain in this area, plus you can see even single axons, neurites, here signals with arrowheads. So these are very powerful immunochemical systems that have given us like very good information on how these nervous systems, at least for this particular ACIL, is organized. We have a similar sort of studies for a different ACIL, but we found that this is not fully satisfactory in the sense that, I mean, as you may know, everybody who's working on comparative anatomy of nervous systems mainly in invertebrates, what they normally use is like antibodies against specific neurotransmitters. The most common, serotonin or fMRamides or some neuropeptides, or you can even use tubulin, okay? So the tubulin is always a little bit complex, like it happens here, because also labels a lot of epithelial cells, okay? So, but if, I mean, but what these markers give you essentially is obviously is just only you see only a fraction of the nervous system. If you use antibody against serotonin, you are seeing only serotonergic neurons. If you are using an antibody against GABA, you're seeing GABAergic neurons. So for us, that was not enough. We really wanted to know how this is really organized. So we want to have access to all the neurons in the animal, okay? So then what we've been doing during the last 2-3 years is taking, oh, well, this is just another way to study neurons and neuron populations and neuron diversity that we've been initiating. This is about to be published now. In Isodiametra pulchra is using single cell sequencing, in which you dissociate the animals and you are able to track the different kinds of or different cell types. And here, for instance, I mean, here are color coded. All of these are neurons of different types. When you sort of like reanalyze this data, you can really get access to all the diversity of neurons and neuron phenotypes within the animal. This has been done for Isodiametra pulchra. We are about to finish for a different ACIL, okay? Anyway, what I wanted to tell you, this is another way of like understanding the diversity, but what we want to know is how all the neurons are located within the body of the animal. And for this, there is only one way to do it. And the only way to do it is by serial TEM sectioning, meaning taking your animal and sectioning ultra thin sections from one very tip of the animal to the other tip of the animal. When you collect and you align all of these sequences and then identify every single cell, then you have a complete map of all cell types and their disposition, of course, within the body. The TEM doesn't select for a particular phenotype, doesn't select for like serotonergic or GABAergic neurons, okay? So what we've been doing during the last 3 years essentially is the following thing. We took a juvenile, which is like a small version of the adult, so it's more tractable. And we've been cutting the first third, been doing sections of the first third of the animal, right from the very tip of the head, let's call it like this, until this organ, which is called an statocyst, is this gravity sensor that many marine animals have. This is about 1/3, and we've sectioned this. We've done about 1,000 sections, okay? Transversal sections of this. We took about 100 pictures per section. Okay, so you get enough resolution. Here you have in the lower part some of these pictures, and have a line, okay, semi-automatically using a series of aligning methodologies. So in a sense, what not me, the student, which is very patient, has done is to take 100,000 pictures representing the first third of the animal at subcellular resolution, has aligned them all, treat them, and then, okay, everything, this is one example of one section, one picture of one section of the animal. The section of the animal will cover more than the whole screen, okay? So every cell is identified by the morphology, okay? This is not automatically done. So this means that student has to be like proficient in identifying cell types, okay? To each one of these cells is given an alphanumeric tag, okay? Saying this is the position in 3D space, okay, cell, whatever. This one blue is like a neuron of that particular type, which is located in that section, in that particular position, okay? So once you have this applied to these 100,000 pictures, okay, and of course the alignment of these sections is complicated, you end up with a map like this one. Here in the lower left, okay, it would be like a lower resolution in which each one of these dots or balls corresponds to a different cell type in the right position, okay? In here, what the student has done at present is to identify and classify 2,000 different cells there, meaning there are still many more. You will see that there is only colors in one side because, I mean, what we've done is analyze only one side of the embryo, assuming that the other side will be a mirror image of the same thing. It's just a matter of simplifying the operation.
Pedro, it's nearly time.
Oh, sorry. Okay, so when this is done, essentially, you end up with a map like this, in which you have different views, color-coded, all the different cell types. You have named here in the lower left, okay? So that's what we have now. We have this, I forget it, this I will skip it, okay? But the important thing is now we have a three-dimensional map of 2,000 cells, aiming at having more, precisely located, okay? This gives us, for us, like an unprecedented view of how the cell types are organized in this animal. Needless to say that we know how different cell types and different subtypes are localized, distributed, asymmetries, etc., etc. Okay? So then I forget this. This is another line of research we are following. But the important thing is, okay, we are analyzing a process like regeneration, etc., which are very interesting. And I wanted to finish with this, is telling you that Oisilomorphs been like very interesting in that sense to understand the evolutionary innovation, in particular the radial to bilateral symmetry, but nowadays are also being instrumental in providing us details on regeneration and other processes of like biomedical interest. Okay, with this I finish. I know that Spaniards like to talk a lot, so thank you very much to all for attending. And if anybody has any question, I don't know how this works. But Adelino, is time for questions, or should we?
Yes, yes. Thank you, Pedro. I think this is a very, very interesting. There is no questions on the chat, but okay. I can ask you one or two questions.
Okay,
one with more curiosity, actually. I probably don't have a complete answer. But so one is, how easy to keep these animals in the lab? Can you keep them or not?
Yes, yes. I mean, there are two species. Okay, one, the second one, which I call it Isodiametra pulchra. Okay, this one we keep it in the lab, and this is easy to keep. The first one, the green color, the one that we get in Roscoff, this can be kept in the lab, but for short periods of time. Okay. Short periods of time, but this is essentially because we are not good enough. I mean, people in Roscoff, which have access to seawater and the right conditions, they are able to keep them for a very long time. So it's a problem that we haven't really worked on the conditions.
Okay.
I mean, yes.
I was going to ask the same question as Ulf Jondelius was going to ask, so I'll let him ask the question.
Okay.
Ulf.
Yes, okay. Well, first of all, thank you for a great talk, Pedro. It was really nice. I was just wondering if you have any ideas why the genome size is apparently so different between Cynoturbella and well, the Oisilomorphs. I think that goes both for dermatids and acils.
Yeah, that's a very good point. I mean, the thing is, I mean, as I told you before, I mean, the Cynoturbella genome is very poorly annotated. Okay, and it's all in very small fractions. So we can give you a real answer. But for acils, what we know is like full of repetitive elements.
Yeah.
So it's a big fraction of the genome is full of repetitive elements, a lot of transposon-like elements. And another interesting thing is like, it's full of islands with no genes.
Yeah.
Which we don't know. Cynoturbella, it's probably has less, but we haven't really, since I mean, all fragments are very small. We have less. Yes, we have a lot of like fragments with repetitive elements, but until we have like a fully, like, or a better aligned genome, I wouldn't be able to tell what are the essential differences.
But is the number of genes similar?
Yes, okay. It's about 20,000. So
there is one more question and then we have to stop here to go to the next one. Ashwin, can you ask the question?
Yes, extremely interesting topic. Well, the green one that you showed from Roscoe is the one that actually has chloroplasts, right? So are bigger genomes also related to the genes that are required to keep these chloroplasts going?
That's also an excellent question. We don't know. We just I mean, we assume that, I mean, it has to be. It has to be because, I mean, there are classical studies in which it's been seen that, I mean, they are metabolic, or they are metabolites that are supplied directly by the algae and are absolutely required for like survival of these animals. But we haven't really studied the genome, the detail in terms of like which genes are present in this genome, which are being taken from the algae, or the opposite way. No, the thing is, there is a group who is trying to sequence the algae. Okay, so probably when we have the algae and the host, then we'll be able to make a story. But so far, no. So far, I mean, we have just assembled.
Okay, sounds very good.
No. But
thank you very much, Pedro. That was excellent. And
thank you.
Now we move on to another topic, quite different, and that's by Tiago Repolho. He's from the University of Lisbon, the Mares Center, and he's going to talk about multi-generational adaptation to ocean warming and acidification. Okay, Tiago, go ahead.
Okay. Okay. Are you already seeing my presentation?
It's fine. Yeah, we can see it.
Okay. So let me just put up the clock to know my time. Okay. So good morning, good afternoon, or good evening. It depends on the case. My name is Tiago Repolho, and for all of those that are not Portuguese, Repolho stands for cabbage in English. And I'm a researcher from Centro de Ciências do Mar e do Ambiente and Faculdade de Ciências da Universidade de Lisboa in Portugal. And I would like to, first of all, to appreciate on my behalf and my co-authors for the opportunity to give this talk. And basically, so without further delay, I will start it. So my talk is entitled Ascend, a multi-generational adaptation to ocean warming and acidification. So following this kind of 5 bullet point slide, this is just an overview of what I'm going to talk about. So I'm just going to provide a short overview of climate change, greenhouse gases, and global warming potential. Then moving up, talk about a little bit of climate change stressors, plus acclimation versus adaptation. Then
sound stopped.
The framework of our project. So basically, climate change, what is basically climate change? It's just the variation of Earth's climate, both at a global or regional level, doing a specific time scale. It can be due to natural processes like volcanic eruptions, or it can be being driven by anthropogenic action, and basically that's where the problem is. So it's the man as usual. The main drivers are greenhouse gases, and basically they are mainly, should I say, 4: the CO2, methane, nitrous oxide, and fluorinated gases. The main sources of these gases, in case of the CO2, results from burning of fossil fuels, electricity production, transportation, forest fires, and land use changes. In case of the methane, basically it's produced, sorry for the word, through livestock production and solid waste landfills. In case of the nitrous oxide and fluorinated gases is basically from industrial production, although we also know that nitrous oxide also comes from exhaustion from transportation. So basically carbon dioxide comprises more or less 80% of the greenhouse gases concentration in the atmosphere, followed by methane 10%, nitrous oxide 6%, and fluorinated gases 3%. In terms of global warming potential, so what is global warming potential, or WP, sorry. Basically, it's a measure of the heat absorbed by any greenhouse gas in the atmosphere. So basically, the larger its value, the more that a given gas warms the Earth in comparison always with CO2 over a time period. For you to have an idea, methane has a GWP of 28, nitrous oxide around 265. And if you go to the fluorinated gases, the ancient CFCs that were banned in 1987, if I'm not mistaken, had a GWP between 4,600 and 13,900. Then the following ones, which were the HFCs, were more or less, in the case, for example, of the HFC 134A, 1,300, and the HFOs have a global warming potential of 4. So moving up to climate change stage stressors, they could act in single or in combination. And when they act in combination, they can have interactive effects. Basically, they work kind of together to produce a result. And that result can be either additive, synergistic, or antagonistic. In case of an effect that is additive, it's when basically stressors head together to create an impact, okay? If they act in kind of a synergistic mode of action, it's basically when these stressors together produce a greater impact than each individually. In case of an antagonistic effect, it's basically when stressors counteract each other. So these climate change stressors, like I told before, single or double or multiple ones, can have effects on marine biota from calcification processes, like for example the sea urchins, photosynthesis in seagrasses, animal behavior, for example, within, for example, cleaning interactions, and also development and reproduction. I'm just going to give you just one small example of a work that we've performed here at the lab back in 2017 with the dwarf seagrass. This is just an example of an antagonistic effect where we expose this seagrass to four conditions: control, ocean warming, and acidification, and the combination of ocean warming and acidification to projected scenarios made by the IPCC to 2100. And as you can see here in this graph, and it deals with the density of the seagrass, the ocean acidification driver or stressor was able to counteract the negative effects made by just the ocean warming. Moving to acclimation versus adaptation. So basically, under this situation of progressing climate change, the marine species have basically three options. They either move, they acclimate, or they adapt. For sessile organisms to move, it's a little bit difficult, if not almost impossible. So they have basically to acclimate or adapt. And that's where it comes the acclimation processes and adaptation processes. So basically, as you can see, the acclimation processes basically involve phenotypical plastic responses, which allow fitness maintenance to cope with environmental change. And they could be kind of three of a kind: reversible, developmental, and transgenerational. Reversible is basically when it occurs within a life stage, and it can span from days to weeks or even months. In relation to developmental acclimation, it occurs when the exposure to a new environment early in life enhances the performance later on. Transgenerational, basically it occurs when the environment experienced by the parents influences offspring performance. In terms of adaptation, it's a process that involves genotypic traits that have evolved through selection, and normally it involves also, of course, multiple generations. Okay, sorry, just have to make a pause. This is a Star Wars slide. I was not able to put the music on, but basically, how did our project idea started? Like Darth Vader said, it was a long time ago, not so long. So in 2014, when 5 researchers from different nationalities, Portugal, Argentina, UK, and Sweden, have attended an advanced course that was named Marine Evolution and Climate Change. And it was held at Christiansberg Marine Research Station, which belongs to the University of Gothenburg. During that course, although it was not just us 5, it was a couple of researchers from PhD students, postdocs, and so on. All the groups, our group was called the Green Group. We all had to do a kind of a 5-hour challenge project. What did this project needed to include? Basically, it needed to include a species that was native, had to be performed at location. The study had to be ecological relevant. Of course, it had to be climate change and marine evolution related. And last, the project had to have a budget below 5,000 euros, because as we all know, research also is quite limited in terms of funding. So following this, our group has presented an idea, which was basically this one on the yellow letters: can acute climate change events lead to rapid evolution in Sea intestinalis? So what did we wanted to do? Is basically to infer if the local habitat history of different Sea intestinalis populations, or these different populations, 2 were derived from pristine sites and 3 from anthropogenic impacted sites, could shape its evasive potential, enhancing ELS stress tolerance to an extreme temperature increase, in this case, a marine heat wave. So later on in 2016, we performed this work, again in Christineberg Marine Research Station. So as I told before, we sampled 5 populations. 2 control ones, we are the pristine sites, and 2 anthropogenically impacted ones. We performed the in vitro fertilization, and then we follow up the development, the early life stage development of Cion intestinalis exposed to two temperatures, control temperature 17 and heat treatment 22 degrees. We analyzed basically 5 endpoints, which was the embryo and larvae development, the settled larvae, metamorphosis, and heart rates. Basically, these are the results of that study. We've noticed regarding embryo development was that the impacted populations presented a lower ratio of normally developed embryos at the increased temperature treatment when in comparison with the control populations. In terms of larvae development, the larvae developed increased at the temperature treatment in all analyzed populations, and the impacted populations, the ratio was doubled in comparison to control populations. Sorry for the time. Okay. In terms of settlement success, we observed a higher number of settled individuals in the impacted populations, again in 22 degrees. And the metamorphosis success of those impacted populations showed a higher ratio also at that temperature. The temperature condition did not affect the metamorphosis success of C. intestinalis on the control populations. The heartbeat rates didn't show any significant overall response to the effect of temperature or site condition. Site condition, so the anthropogenically impacted and the pristine. Basically, our conclusions were that the temperature had a significant overall effect of the analyzed endpoints, with the exception of the fertilization and heartbeat rates, and that Cion intestinalis ELS from anthropogenically impacted environments were negatively from control, environments were negatively affected by the high temperature conditions. Also, the populations from the anthropogenic impacted sites show an overall consistency in their positive response to the simulated heat wave event. Basically, our study sustains the hypothesis of a species-specific pre-adaptation in organisms, in this case, the ones that originated from anthropogenically impacted sites. And this leads us to conclude that most possibly these populations could be more resilient to future heat wave scenarios in comparisons from the ones that derived from the control sites. This work right now has been accepted, just a small information at the GAB, it will be under gold access. So if later on, if people can check all the details. So as that yellow line says, this work gives us some ideas, which takes us to the Ascend project, okay? So our project basically has 2 main objectives, is to check the ability of C. intestinalis to adapt or not to climate change variables, in this case, ocean warming and ocean acidification conditions, and to check how the potential or predicted interactive effects may or may not be reflected on biological responses of our biomodel within a long-term multi-generational context. Why Cion intestinalis? Basically, from the work that we've done following the work that we've done in Sweden, this organism is easy to be captive bred. Also, it has a short generation time, and the early life stages, the Cion intestinalis early life stages are thermal sensitive, and also because of available genome-related resources. From an ecological point of view, mainly it deals with the invasive potential of this organism, although it's native here in the northern hemisphere, and also because it's an ecosystem basically engineer. So it could kind of make or produce problems, sorry for my English, due to species competition, and also because it's a strong colonizer of free substrate. Also, this organism is also kind of an economical engineer. So it disrupts a lot of human activities, especially some dealing with the aquaculture. And we have a special case here in south of Lisbon, in Lagoa I think it's Lagoa de Óbidos, the name, Lagoa de Óbidos. With mussel farming, when during these last 5 years they had a mussel production reduction of nearly 90% due to this organism and another invasive tunicates. So to do this work, we've performed or we've collected populations from 2 sampling sites south of Lisbon, like I told before, and also from the Rias Barras in Vigo, where they had the support by a couple of people from HECIMAT, the Estação de Ciências Marinhas de Orelha, and these populations were brought to Laboratório Marítimo da Guia to perform the laboratory acclimation, and following this, I'm presenting our experimental evolution design, the idea that we had to do. So basically, the populations were kept under a common garden, how do you say, common garden, yeah, yes. Common garden condition, sorry, and where they were kind of bred together and were able to reproduce into several generations. Subsequently, the idea is to replicate that common population into different lines. In this case, the control one, the ocean acidification, the ocean warming, and the combination of ocean warming and acidification. And at specific times of this experiment, we performed basically reciprocal assays where we test for adaptation and check the correct response of all the treatments in relation to each other. Okay, our project basically has 5 working packages. The first one was related with the collection of organisms and acclimation, and so we basically have these 4. And these 4 are, the first one is, deals with development and reproduction, the third one with physiology, the fourth with gene expression, and the fifth with neurobiology. Okay, but that we had, since we collected these organisms in the wild, we had a couple of problems that had to deal with sustaining the populations under laboratory conditions, and the inability to obtain mature adult individuals in the lab. And adding to that, we had a visit from a friend of ours, sorry for saying this, and like the French say, nous avons le plaisir de rencontrer notre ami Corona, which with the restrictions that were imposed in doing the experiments here at the lab and in our country and all over the place, led us to stall the using Cion intestinalis and moving to another biomodel, which is an amphipod species, Gamarus locusta. It's a species that we already have for a couple of years experience with in breeding them in the lab, And it's a rarely species. It possesses sexual dimorphism, and it's a gonochoric species. And it's quite easy to be captive bred. This was a short movie of one of the aquariums downstairs. And also it has a short generation time. Our new approach, we will be performing, because due to the uncertainties dealing with this pandemic situation, we will move up to
There's problem with the communications. I think we lost Tiago. Tiago? Give him 1 minute, see if he comes back. Okay, so anyway, I think he was getting to the end of his talk. If he comes back, we can ask him some question. So we move on to the next talk, and that's by Emanuela. Let's check here. So Emanuela Dattolo from Stazione Zoologica and Tondorn in Naples, Italy, and she's going to talk about circa grass, adaptation of the seagrass circadian clock to latitudes. So Emanuela, please go ahead.
Good afternoon, everybody. Can you see my screen?
Yep.
Okay.
Not yet. No, you haven't shared the screen yet.
I shared it. Okay, I will try again. Condivide. Now? It's okay?
Now just click.
Okay.
That's it.
Okay, good afternoon. I will show you the first results of the project Circagrass, adaptation of the seagrass circadian clock to the latitude. This project was carried out in 2019 in collaboration with Stazione Zoologica, Sissimar, and Gothenburg universities. Seagrass distribution is strongly mediated by the extent to which multiple interacting variables remain within suitable condition, and among those, the climatic variables such as temperature and solar radiation are the most important factor for the geographical distribution of the species. And so the current distribution of the species that worldwide that you can see here reflect the requirement in terms of temperature and solar radiation of the different species. Along the coast of the North Atlantic Ocean and the Mediterranean Sea, the most important abundant species are Posidonia oceanica, which is also the endemic species of the Mediterranean Sea. But there are also Zostera marina and Cimodocea nodosa which spread across the Atlantic coast, Zostera marina in the northern portion and Cimodocea nodosa in the southern. And there is also Alophila stipulacea, which came from the Red Sea. So climatic model and several observations indicate that the global and regional climate warming are causing a fast geographic shift of the isotherm over time, and also a shift in the seasonal timing of temperature. So these factors are affecting the global distribution of seagrasses, and a mechanism by which organisms and also plants try to escape to global warming are the shift in their biogeographic range, moving the distributional area in the northern sites. But this is not possible for all species for a different kind of factor. For example, for Posidonia is not possible because there is the European continent. while for Halophila was possible, and then the species is present also in the Mediterranean Sea. But there are also many intrinsic factors related to the sensitivity to the species, to the photoperiod, for example, that can avoid this shift in the expansion range. So a crucial point to assess how seagrasses will be distributed in the future is the study of the photoperiodic sensitivity of the different species. Photoperiodism refers to the ability of the organism to adjust the physiology and to the seasonal change in the environment according to the length of the day. And this is made possible by the circadian clock, that is an endogenous timekeeper system, which is able to track the external signals, mainly from light and temperature, and is able to orchestrate the rhythmic expression of several genes, and which are related not only with the light, but also with many other key processes for the plants. So, from a molecular point of view, this system is based by a transcriptional translational feedback loop, in which a clock gene is regulated negatively by the protein, by its protein. And the plant clock are composed by at least 3 interconnected loop, and by the activation and deactivation during the day of the genes related to this loop, plants are able to orchestrate the activity of their metabolism, not only according with the external signals, but also according to the internal necessity of the plants. So, not only external environmental cues are able to interplay with the clock and regulate the expression of circadian clock genes, but also post-transcriptional RNA modification can regulate this system. In particular, epitranscriptomic, that are chemical modification of RNA molecules, and then they are able to modify the molecules without to modify the sequence, are known to have a role in the clock of several organisms. And in particular, the M6A RNA methylation, that is also the most common type of mRNA modification, is known to be a role in the light-night regulation of the clock in mammals, and also there are few work on several plants. This modification is established by 2 kinds of genes, the writers and the eraser genes. Writers add this amethyl group on the mRNA, and eraser remove this group. By the concerted work of these 2 kinds of genes, the messenger, the mRNA life is regulated, and then this regulation has also an effect on the daily regulation of plants. So the aim of this project was to investigate the adaptation of photoperiodic sensitivity of different seagrass species and population along a latitudinal gradient. And in particular, we had 3 objectives. The description of the epitranscriptomic regulation of the circadian clock system between 2 species, Cimodocia marina, Zostera marina and Cimodocia nodosa, and between population of Zostera marina collected at the boundary of the distributional range of the species. Then we would like to define the daily expression pattern of genes related to the light response and energy metabolism in both species to see if there are differences in the usage of energy of the plant. And then the other goal was the identification of genetic polymorphism in the species Cimodocia nodosa, looking at the polymorphic at the SNPs in population collected in along the distributional area of this species. Regarding the gene expression analysis, We collected the sample during a 24-hour cycle, and in Faro, in Portugal, we collected plants 6 times during a day, and at midday, midnight, sunrise, solar nude, and also at dove and sunset, where the in correspondence with the change in the transition between light and dark and light. While in Charno, since there are no dusk and dove, when we did the analysis, we collected the sample in at 4 times during the day. In both cases, the plants were really shallow, and that the light that the quantity of light was comparable. For what to investigate change in M6A modification and how this change can affect the clock of seagrass, we decided to follow the variation in the global level of M6A in the transcriptome of the species, and we follow also the expression of genes related with this modification via real-time PCR. The results of this work are published, and I will give you an idea about what we did. Before to start with the real-time analysis, we performed an inventory of the genes related to this process in seagrasses using the genomic and transcriptional data available, and we compared the genes with the genes of Oryza, sativa, and Arabidopsis. It is to identify if seagrasses possess the whole repertoire of the M6A genes. And I won't go in detail in this topic, but we identified genes for all of this gene family, and also realized that in seagrasses, some of these family has a lot of transcript, redundant transcript in comparison with land plant. And we are investigating also if this redundancy has an adaptative significance. So from this preliminary analysis, we performed the real-time PCR. And we did the analysis on 2 writers gene and 1 eraser gene. And as you can see here, we obtained during the day a clear rhythmic pattern of expression. And this pattern of expression show that for both species and for all of the gene analyzed, there are the maximum peak of expression during the dark period of the day. And this, the same pattern with the peaks in the day was also observed in the percent of M6A measured on the transcriptome. So, but if you see the profile of the 2 species, Cimodocia nodosa in orange and Zostera marina in blue, there are an asynchrony between the peaks, and for all genes, Cimodocia show the maximum at midnight, while Zostera early at the sunset. But it is the contrary for M6A. This is the same analysis, but with the same gene and again the percent of M6A, but performed using the population of Zostera collected in Faro, that is in blue, and in orange in the population of Charno. In this case, there are only 4 time points, and you can see that there is a perfect match between the expression profile of the population of the 2 latitudes. So, in conclusion, what we observed in this analysis is that there was a strictly rhythmicity in the M6A transcript, and this rhythmic was also similar to what observed in the level, in the general level of M6A in the transcriptome of the species. And we observed, in any case, the peak during the dark period, and it could suggest that the regulation of circadian clock by the RNA, by epitranscriptomic mechanism is mainly activated during the night. We also observed an asynchrony in gene expression between species at the same geographic location and a similar pattern of expression between populations of the same species. This could suggest that the regulation of the expression of these genes is under an endogenous control, and it could be also related to the difference of the two species in the use of the energy during the day. And then we can conclude that M6A methylation participates in the daily regulation of gene expression and the circadian function of seagrasses, and this regulation could affect the photobiology of the species and the ability of the species to spread across different latitudes. So regarding the other task of the work, the analyses are ongoing, but I will show you the first results that we obtained. For example, for the daily expression of genes related to light and other processes that we know are regulated in a circadian manner. This is a panel of genes that we analyzed, and then we are analyzing genes related with the sucrose synthesis and transport, photosynthesis, light harvesting, and also photoreception and circadian clock. And we are trying to obtain a daily, we are trying to identify which are the expression profile of these genes and assess if these genes are related with the light, are regulated with the light. And as you can see from this PCR, this is true, at least for Cimodocia, because all genes that we analyzed cluster really nice with the different light phase of the day. And in particular, this PCR showed that along the first axis, which explained more than the 50% of the variation in the gene expression of these genes, there is there are the night and there is midnight and solar noon. Then they do extreme, the point with the maximum difference in the light, but also the second axis is clustered really well with the end of the day on the left and the start of the daylight in the right. We are doing the same, the idea is to compare the two species to see if they follow exactly the same pattern. And as these are, for example, the same genes that we have already analyzed for Zostera marina. And if you see the profile of the sucrose synthetase, you can see that between the two species, there are clear differences in the expression profile, because in Cimodocia, these genes peaks in the early morning, while in Cimodocia at midday. And the same gene analyzed between Zostera population also show difference in their expression profile. And this suggests that the species could be able to change the daily expression of these genes according to the energy available and the physiological necessity of the plants. The third objective was to identify the genetic polymorphism that could be related with the latitudinal climate. We performed this analysis only in Cimodocia, and for this analysis, we collect sample in Gran Canaria, in the Mediterranean Sea, and in Faro, which is also the northern limit of the distribution of these species. The DNA was extracted in Naples, but the two-birad analysis was performed in Gothenburg, and then we are analyzed there the data. after the sequencing, we were able to identify many SNPs, and this is just the first analysis that we performed to see if in the SNPs that we collected, the SNPs that we detected, there are some outliers between population, and we were able to identify 14 outliers genes. And other analyses are under, we are doing many analyses to try to assess if these SNPs are also related with genes related to the photoperiodic sensitivity. Thanks a lot. And these are also the other researchers that are working on this data.
Thank you, Emanuela. There are some questions. So Sylvia, you can ask the question.
Sylvia? Can you hear me? Can you hear me?
We can hear now.
Okay, I'm sorry because my microphone doesn't work so well. So I'm wrote my question on the chat.
Yeah, that we can hear perfectly.
Okay, so nice question, nice talk, Emanuela. Okay. So my question is: there's some evidence on the protein level of erasers on the writer's genes along the daily cycle, you know.
I don't know because we never see the protein content of the plants. Then we just tracked the level of the transcript. And from literature, I don't know.
Okay, nice, thanks.
Okay, before I pass to Davide, I'm going to ask a question myself because it's a different kinds of question. So my question is basically, as you seem to have mentioned in one of your slides, there could be some like metabolic underlining rhythm, you know, because you've got respiration, photosynthesis, and so on. So did you try to put the plants under constant light or constant darkness for some time to see if these rhythms are maintained or not?
This is me and Miriam Rocco, it is the co-PI of this work, are proposed this analysis for our second assemble project, and we will. We hope to be able to perform this analysis in the future because, of course, it could prove that there is a real circadian regulation in this process. And then I hope to have the results in the next years.
Okay, so that's why now Davide wants to ask questions because he wants to know what you are asking next. Come on, Davide.
Yeah, so the first question is about this rapid transfer between Faro and Christinenberg. Does that involve a lot of preparation for the technician in these two institutes, or did you have you handled that with your research team without any problem? So was there required a preparation before your arrival?
No, we prepared me. Because, for example, for the tuberad analysis, we prepared the sample in Naples, and but we arrived in then Davide, we were not in Christinenberg but in Charno.
Okay.
Okay, and the only what we did before was just to prepare the kits for the analysis, and then when we arrived, they are ready for help us, and it was really nice because we spent 1 week to do both the analysis for the tuberad, and during the day and the night, we also did the sampling for the gene expression analysis, and it was really fun.
Okay. And the second question is that if you plan to add, to have samples or to visit some other geographic area to, let's say, enhance the sampling, or is it fine as it is now?
I don't know if it is possible because looking for it will not increase the latitudinal gradient of the analysis could be really nice. I don't know if it's possible for the presence of the absence of the station of the Mari research station in the area that we need. For example, for Cimodoce annodosa, the original plan was to try to use a sample from Mauritania for the SNPs analysis in order to catch the sample, the genotypes of the southern limit of the species distribution, but it was not possible, for example.
Okay, thank you very much. Before we move on to the next speaker, I just want to ask Tiago if he has any concluding remarks that he wants to make, or if it's okay. And if there is any questions for Tiago, because he was thrown out of the conference.
I'm sorry, but from time to time, it also happens that maybe some kind of digital coronavirus also steps in the lab and ravages our connection. But I don't know where I stood on the presentation, but the final remarks basically were that besides the acknowledgements, of course, we are moving up to using a new biomodel species, which is Gamarus locusta, is an amphipod species.
Yeah, we could hear that part.
Yeah. Okay. And basically, we already have 3 generations of those organisms here at our lab under common garden conditions. The conditions was the word that skipped me a couple of minutes ago. And we are basically going to perform 10 generational acclimation on these organisms, also from a multi-stressor point of view. What do I mean by multi-stressor? From the combination of ocean warming, ocean acidification, and or hypoxic events, and maybe most probably also to deal with the extreme weather events like marine heat waves.
Okay.
The acknowledgements are just for Assemble, okay? Also for Sam Dupont, my co-author for my Swedish paper, and also all the persons from ESIMAT that really gave us a huge help in collecting the populations of Siona in Rias Barras de Vigo. And I'm sorry again. Sorry again.
Thank you very much, Tiago. I don't see any questions on the chat, so I will.
Your microphone, Adelino.
Yeah. The microphone.
My microphone is down, sorry. So I was going to say, because since there is no more questions, we'll move on to Maria Rita Caracciolo from Station Biologique Roscoff. And she's going to talk about metabarcoding analysis to explore proteus phenology and diversity. So go ahead, Maria Rita.
Hi, everybody. You can see my screen, I guess.
Yeah, we can see. You can just click for presentation. That's it.
Yeah. So thank you for inviting me to participate to this conference. I am Maria Rita Caracciolo from Roscoff Biological Station. And I will present you here the results from my PhD studies that regards metabarcoding analysis and the study of temporal patterns of microbial communities. The main objectives of my studies are in fact to detect and describe the temporal patterns that are owned by those microbial communities, and to understand which are the processes that govern the structure and the seasonalities of those communities. Because as we know, we can go towards 2 directions to understand better these processes. One direction is abiotic factors, and so the environment, and on the other side, we have biotic interaction. In any case, it's important to study the whole figures of the temporal patterns, because as said this morning in the presentation by Chris Bowler, those communities are very important for our ecosystem and for the ecosystem services that they provide to us. They are at the base of the food chain, they produce oxygen, and they participate in many biogeochemical cycles. So in general, plankton and phytoplankton is known to have an annual cycle of biomass that change in biome to biome. So let's concentrate a second to the temperate biomes. And we know that here, plankton is, annual cycle of plankton is mainly structured by the water mixing and the stratification regimes that are created at a certain season, in summer in particular. So following this annual cycle of biomass, we have, of course, different succession of groups. For example, in the temperate biomes, it's known that the diatoms are the group that bloom normally in spring and in autumn. But when we go inside each of these groups, we also have a succession at the level of species, different species that succeed in every season. So on one side, we know that some of the parameters that are most important for plankton, because most of the organisms are photosynthetic, is light. And for this reason, we took light parameters in consideration, like photosynthetic active radiation or the coefficient of attenuation for 90. But aside with light, what has been brought by literature in the last decades is that temperature is one of the most important factors, sometimes also more important than light, because it has been shown lately that many species are going towards a North Pole because of the climate change and the change in temperature. Then, aside from light and temperature, we also have to consider nutrients, macronutrients, of course, because as I said, these organisms are participating and shaping the biogeochemical cycle as much as biogeochemical cycles are also influencing those organisms. So nutrients have to be taken into consideration. But aside by environmental parameters, what it has brought to light in the last years is that biotic interaction have a very important role. Since that, we know there are some special community assembly rules, so some species can coexist each other while some others simply cannot because they enter in competition. So we know that there are different kinds of interaction in the ocean, from symbiosis to parasitism, arriving to predation and mexotrophy. They can be both positive or negative interaction, depending if these organisms can coexist or not. And it's important to study them as much as environmental parameters, because lately we understood that biotic interaction are also influencing those temporal patterns. So what we do this kind of study with time series, and our time series station is in the Western English Channel, Somlit-Astan, and is operating twice, our team is operating twice a month to collect samples. So on one side, we have morphology, 10 years of microscopic counts that are bigger than 20 micrometers and that are able to catch the microphytoplankton. But also what we do is to extract this DNA, amplificate it, sequencing, and so create metabarcoding. And metabarcoding data are 8 years for what concerned the V4-18S-R DNA. We have two size fractions from 0 to 3 and more than 3 micrometers. And these allow a comparison with the morphology, because this method, traditional methods that are microscopy, for example, are of course limited. With this kind of method, we can study just size fraction. Only the biggest mesoplankton, macroplankton can be seen by human eyes and by microscope. So it's important to integrate these other methods, metabarcode methods, because they allow us to access all different size of organisms, smaller organisms like nanoplankton, microplankton, and they also allow us to reach a much higher diversity, because we are talking about millions of reads. In fact, when I compare diversity in my two data sets, we can see that diversity is much higher in metabarcodes data, because of course for morphologic data, I just have 146 species, but for metabarcodes data, I have more than 25,000 OTUs. And this increases, of course, the diversity. Diversity is also specifically higher in winter. This is also another fact that is known, because in summer, normally community is composed by few species, that are able to bloom in a very abundant way. But instead, in winter, where the conditions are a bit limited, temperature is lower, we have much more species that are able to flourish in the water. When we studied beta diversity and we compared, again, the two datasets, we have a similar results, and we can see that the community is able to come back at the same composition every year, meaning that along the 8 years, our community is very resilient. But who are those guys in the water? So by studying the morphology that I didn't show here, we can only see mainly diatoms. So that's why it's much more interesting having metabarcodes. Here we can see that, of course, the most abundant are diatoms and dinoflagellates. But we also have access to all the other smaller groups, heterotrophic, parasitic, and symbiotic groups like Cercozoans, Ciliophora, Picozoa, Sagenista, fungi, Syndiales, and all of those groups that are overlooked in most of the studies exactly for a problem of size. But another problem that we have in metabarcoding is also that there could be many biases that are that could be, let's say, make us overestimate some groups. In the case of dinoflagellates, for example, we know that they have a big DNA that can amplify better, and so the dinoflagellate could be an overestimation. But we also know that in the coastal environment, dinoflagellates are very, very important. So it could simply be a true result that dinoflagellates are also more abundant than diatoms in this station. But the biggest problem that we have with metabarcodes is about the assignation. As you can see in my slide, most of the names of some of the most important groups are not clear, because when we use databases, not always we can arrive to a good assignation. In this case, we use the peer-to database, but of course, it's important for us to arrive at least to a genus or a species level for those groups that are the most important and dominated in the water. That's why we assigned manually through BLAST the sequences that were for us the most abundant. But also what we did, thanks to the Assemble Plus project, it was to have a more precise assignation of the diatoms. In this case, what we can see by comparing diatoms from morphologic and metabarcodes is that benthic diatoms are very abundant. In fact, our system is characterized by a mixing of the water all over the year because of the tide that is present and characterize the ocean regimes. So of course, benthic diatoms are brought into the surface because of this mixing, and they are very abundant for this reason. But what we can see also, thanks to metabarcoding data, is all the nano-pelagic diatoms that are overlooked with the other methods, and that are also more abundant than the other diatoms that are present just in the morphologic data. So those sequence of nano-pelagic diatoms, being the most abundant, of course, is very important to assign them in a more precise way. That's why we firstly built a reference tree of diatoms in which we would like to place those sequences in the future to have a better assignation. But for now, this tree has served just as a reference tree in order to correct our manual assignation and to correct also all the sequences that are present in GenBank that sometimes are wrongly assigned. But the most important things that we reached, thanks to our colleague of Naples at the Antodon station, it was to arrive to assign to arrive to learn those techniques that are able to concentrate, that are needed to concentrate the diatoms through stools. So all those techniques that regard the acid concentration and the speed of centrifugation, that allowed us to bring this technique back in Roscoff with us, because we had some of the sequences, some of the strains that we had that were corresponding 100% with our sequences could not be perfectly assigned. So thanks to this technique that we brought, and thanks also to the use of the infrastructure of the TEM at the Anton Dorn station, we were able to assign mini-discus variabilis, for example, that is really the most abundant species, and Arcocellulus, and many other also Thalassiosira species that I'm not showing here. What we did, why infrastructure access is useful is also because we can access the data. And thanks to the access to the data, the metabarcoding data of Naples, we could compare the diatom diversity at the two sites, Roscoff and Mare Chiara station. So while on one side in Roscoff, we can see the diversity is very constant along the year, in Naples instead is variable. And I would have expected the opposite, for example, to have a more variable diversity in a mixed environment respect to stratified environment, like instead is the Mediterranean Sea. Also, what we did was to compare the OTUs that are driving this succession. And we could see that, of course, the OTU are different between those two environments because they are very different environments, but some of them are in common, like, for example, Thalassiosira concavuscula. Even though these results need to be confirmed with the new OTU table that we are building, because what is needed to compare two different data, metabarcoding dataset, is to have same procedure of building the OTU table and of assigning those sequences. And this is an ongoing project that we are pursuing now. But coming back at the temporal structure of my communities, what we were able to do also, it was to detect the temporal patterns very precisely thanks to the DBMEM models. Basically, we were able to do an RDA analysis between the DBMEM and our community and to extract the main temporal patterns that are all concerned with seasonality. In the first axis, we could see the winter-summer seasonality, while in the second axis, we could see the spring-autumn seasonality. And in the third axis, instead, we detected a sort of biannual pattern. And thanks to the DBMEM, we were able to explain more than 50% of the variance. So what we did also was to try to understand which were the environmental parameters connected with those temporal patterns. So the winter, summer in the first axis and the spring, autumn in the second axis. And so we use a Spearman correlation with between the environmental parameters selected and the RDA axis. In the first axis with the winter summer seasonality, we could see that most of the correlation was for the light related parameters like photosynthetic active radiation, temperature, and macronutrients. The same kind of correlation were detected in the second axis, but of course not strong as much as the first one, that is the one that explained most of the variance. And the third axis, we were thinking that it could be related to the tide amplitude because of this biannual pattern. And in fact, we were able to find a tiny correlation with the tide amplitude, with the nitrate. But these results, of course, have still to be better checked. But what we did also, it was to calculate the variance partitioning between our explanatory variables, so our environmental parameters in this case, and the DBMEM that are representing the community dynamics. And I'm going to explain you soon exactly what are those community dynamics. What we can see here that in blue, the environmental parameters alone, they were explaining 28% of the variance for both datasets. But the community dynamics, the DBMEM that we used, they were explaining instead 44 and 48% of the variance respectively for morphologic and metabarcoding data. What does it mean, all this? So this means that our temporal structure, of course, they are related to both abiotic and biotic parameters. But on one side, the abiotic parameters, the environmental parameters selected, are more able to explain those temporal structures that happen in a broad scale, in a larger scale. And the ones selected and confirmed, like we already knew from the literature, were photosynthetic active radiation, coefficient of light attenuation, temperature, followed by the macronutrients, phosphate, nitrate, silicate, and other parameter related to the biogeochemical condition of the water. But most of the variance, as I told you, was explained by the DBMEM, and the DBMEM are representing the community dynamics. Basically, all the dynamics that are created by the species themselves, by autogenic processes owned by the species, like, for example, interaction, reproductive dynamics, all other random events that can happen. And all of these community dynamics are able to explain the temporal patterns that happen in a finer scale, so in a smaller scale respect to the other environmental parameters. So in conclusion, we were able to describe specific temporal patterns and to demonstrate that they are repeatable in time. So we saw that the community is very resilient and that the main temporal patterns, it was the seasonality. Then we were able to confirm the environmental driver of the cessation that are light-related, temperature, and macronutrients, as it was, as we studied already in the literature. And also what we are doing in an ongoing project is to study better the interaction between organisms with the co-occurrence networks in collaboration with Nantes, the University of Nantes. And we are concentrated also on the keystone species and on the core and transient species, because every population can, all populations can be divided into core and transient. So some of these are core, meaning that they are present all over the year. Some other are transient, meaning that they are sort of tourists coming and going when they want. So this is another ongoing project. And also another collaboration is about ecological indicators in order to study better the impact of anthropogenic disturbance. And this is of course important for biomonitoring studies. Thank you for your attention, and I let you with this beautiful picture of Roscoff.
Thank you, Maria Rita. There's a question. So please, Yubehu, can you ask the question?
Hi, Maria Rita. Can you hear me? Sorry.
Yes. Yes,
okay.
you can talk. You can talk.
And I'm sorry if you already mentioned it in your talk, but I'm just wondering if you also looked at the metabarcoding data for protists that has sizes larger than 20 micrometer and combine it with microscope observation.
Yeah, the whole point of my study is this comparison between morphology and metabarcoding. Metabarcoding, though, is smaller than what you said, is between 0, 2, and 3, and between 3 and 20 micron. More than 20 micron, then we go already into morphology. And yeah, we also have other metabarcodes with different gene marker that are able to catch metazoans and protists, but we have just 3 years of those. That's why we decided to use just the V418S rDNA that can be very well compared with morphology, because we have almost the same years, 8 years and 10 years.
Okay, thank you very much.
Yeah, I've got a question. Are you going to do any kind of genomics? Because yeah, it seems to me if you want to compare times and places, like we saw this morning, you may learn quite a bit about what they are doing, because you, like, for example, you're comparing Naples and Roscoff.
No, so yeah, that it was in the project of Assemble. We wanted to compare these two environments to show that when we talk about most abundant diatoms and when we talk about the diatoms that are driving the succession, of course, those are different between different places. And Roscoff and Naples, they were good places to compare, because in Roscoff we have a mixed environment, so all over the year the tide it's mixing the whole environment, while in Naples at Marichera Station in Mediterranean Sea there is a stratification. And so I was curious to see how the temporal patterns are changing and how the OTU that are driving are changing between those two environments that are so different. But the main point of the collaboration with Napoli has been, in fact, to brought home these techniques that they were well managed there in order to better assign the most dominant sequences.
Okay. Anyone, any more questions? It seems as if no more questions. Well, thank you very much. And I also like to thank all the other speakers and the audience. So tomorrow we'll come back at, let me just check here, 11:30 Central European Time for presentations from 3 marine stations. And then we follow with talks again in the afternoon. So thank you very much and see you tomorrow.
At NHBS, our purpose is to support those who are passionate about wildlife, ecology, and conservation. We stock a variety of books and equipment to suit the needs of marine conservation professionals, and our innovation and research team are here to help develop custom products for any project. We are happy to provide advice and to support you before, during, and after your purchase. Visit nhbs.com today to find out more.